Ladies and gentlemen, sorry to interrupt your coffee. Please go ahead and enjoy it and enjoy your dessert. I think we'll just start the evening's program. So my name is May Akrawi. I'm the Consul for Science at the British Consulate General here in Houston, and along with Catherine Santamaria, represent the UK science interests here in the 6 states of the Southwest in the US. We're part of a Foreign Office team of science attaches, about 100 of us based in about 45 embassies and consulates around the world. And what we do is exchange best practice in the countries we're at on science policy and facilitate research collaborations. And I think I can safely say in Texas we've had a lot of interactions on both, and there's a lot of great science going on here, a lot that has happened through the Texas UK Collaborative and beyond. And on the policy side, we've been very fortunate working with Dr. Neil Lane and Kirsten Matthews here at the Baker Institute Science Technology Policy Program for many years. And in fact, this is our 4th in series of stem cell policy and research programs. And we really appreciate that partnership. We've also worked with the Baker Institute on climate change, on nanotechnology, and other science and technology policy issues. It's a subject that's dear to our heart, dear to Dr. Lane's heart, about how impartial and the best science advice is given to policymakers so they can make their decisions in an informed way. And that's really been the basis of a lot of our work together. And we also feel it's very important to engage the public on science, and this is one of the great fora that we've been able to do that through the Baker Institute. So we appreciate a lot of the scientists being here, but also members of the public and the legislative members, because it is important to engage the public on issues that affect them. And in the UK, we've gone through many versions of this, from the public understanding of science when I was a student many moons ago, where scientists try to get better at explaining science to members of the public, to the public engagement of science, where there is real debate, there is real input from the general public into science policy. So without further ado, I'm going to introduce our first speaker. Our first speaker is Dr. Malcolm Gillis, who I've had the pleasure of knowing since I've started my job, when he was the president of Rice University, and he was in that position for 10 years. He is now the Irvin K. Zingler Professor of Economics at the Baker Institute and Executive Director of the Texas UK Collaborative. And Dr. Gillis was very instrumental in starting that collaborative in partnership with the British Consulate General here. And he has distinguished career, started at Harvard and Duke, and his bio is beyond what I can read in this 5 minutes, but Dr. Gillis was also chair of Biohouston, and I also want to mention something that's relevant to what's been going on these 2 days, in that in March 2008 he was appointed by the governor of Texas to serve as vice chairman of CIPRIT, the Cancer Prevention and Research Institute of Texas, and this is a $3 billion effort over the next decade, which I'm sure Dr. Gillis might say a couple of words on. And in that effort, he's also been a great partner in reaching out to the UK and trying to make sure that the best scientists from the UK and Texas are collaborating on cancer as we do on other subjects. So, Dr. Gillis.
Well, good evening. It's my pleasure. It's my second appearance before some of you today, so I'll try not to put you to sleep, but I'm going to be very short this time. But we owe thanks to a lot of people. I'm going to mention some at the end, but I certainly want to thank the Baker Institute for clearing the schedule so that we could meet here. Ed, thank you very much. The Texas UK Collaborative is now entering its 8th year. The collaborative had its origins in my visit, arranged by the British Consulate General in Houston, to Edinburgh and London exactly 8 years ago tomorrow. This was immediately after 9/11, and Elizabeth and I left for Britain on the first transatlantic flight permitted after that terrible tragedy. Arriving first in Edinburgh, where I had to give a talk to the Royal Academy, we were greatly cheered by a very thoughtful post-9/11 editorial in the Edinburgh paper, The Scotsman. The editorial asked, and you know the Scots love to brag about their inventions. My people are Scots, I can say this. And the editorial asked, what was Scotland's greatest invention? You know, anybody know? Anybody know? The answer given by the Scotsman in the editorial was Scotland's greatest invention was America. And that was because more than a third of the signers of the Declaration of Independence were of Scots heritage. They're not shrinking violets, as you know, as a people. After Edinburgh, the British Consulate General arranged a meeting with the then Minister of Technology, Lord Sainsbury, who was the most remarkably well-briefed minister I have ever met before or since in my life. And after an hour and a half, I walked out of his office knowing that the collaborative had just been born, together with a commitment from him for several £1 million to support the British side. The collaborative, under the very able executive leadership of Dr. Dennis Heaton, has flowered ever since. We've been written up in Nature and other periodicals as a model for international scientific collaboration. The collaborative has grown from just a few universities in Britain and the U.S. to 20 universities and medical centers, 11 in Texas and 9 in the UK. And please permit me to proudly call the roll of these schools. First in Britain, Imperial College, London, King's College, London, Queen's University, Belfast, Swansea University, University College, London, University of Cambridge, University of Edinburgh, University of Southampton, and University of Strathclyde. In Texas, Baylor College of Medicine, Methodist Hospital Research Institute of Rice, Methodist Hospital Research Institute, Rice University, Texas A&M, University of Houston, University of Texas at Austin, University of Texas Health Science Center at Houston, University of Texas MD Anderson Cancer Center, and University of Texas Medical Branch in Galveston. That's a formidable list of institutions, and I am pleased to have every one of them as members of the collaborative. 14 of these institutions, including 6 from the UK, are presenting today and tomorrow at this conference. And also we have distinguished speakers from the Medical Research Council and the UK National Stem Cell Network Steering Committee. So I'm going to close by thanking all present and all possible future funders of this path-breaking collaborative venture. So far, the largest funders have been the British government, the Farish Foundation here in Texas, and several private donors in Houston. So now I hope that I have performed my major function, and that is to warm up the crowd for Paul Lynch, Neil Lane, and Lord Patel. Again, welcome to Houston, and to Texas, and to Rice.
Thank you, Dr. Gillis. Now it's my pleasure to introduce my boss, Paul Lynch, who's our Consul General in Houston, and he's been in this position since 2007. And not just because he is my boss, but Paul has really been supportive of our science, technology efforts throughout his stay here, and a great supporter and very active in the climate change, both science and policy in Texas. And Paul has had a long career in the civil service, so he's had several policy jobs in the UK, from the Home Office to Cabinet Office, and then served in Tokyo for many years as First Secretary of Science and Technology, so he knows very well the role that Catherine and I are trying to do here, which is great. And he also took full-time language training in Japanese. So Then that was very handy because Paul worked in the commercial section at the embassy in Tokyo as commercial consul in Osaka, consul general in Osaka, and then he joined us here. So without further ado, I'll introduce Paul Lynch.
Thank you, May. It's a real pleasure to be here. My remarks also very short. My job is really just twofold. One, to welcome you on behalf of the consulate and the British government, and two, to say thank you to a number of people without whom this would not have been possible. Before I do that, I just want to say a couple of words about this area of stem cell research and genomic medicine. We're very proud in the UK of the work that we've done. Not only the scientific work, but also the policy, the ethical considerations, and surrounding areas, which are so important to the development of a scientific field. We've spent many years discussing, surveying, debating the landscape of this topic with academics, professionals in the field, religious groups, faith groups as well, and the general public, including contributions from the major organizations that we set up, the Human Genetics Commission, which is an advisory commission, the Human Fertilization and Embryology Authority, which sets the regulations, the science committees in both the House of Lords and the House of Commons, House of Lords represented here by Lord Patel today. And I think it's a model of consensual policy making. As somebody who spent his life working in government and knows how difficult it can be, this area is one where the governments of all hues, scientists and others, have got together and really done their best to put together the policies which work without party political favour or bickering. And I say that especially at the moment as an encouragement to those in the United States that it is possible, even in healthcare, even in this most difficult of topics, to find a consensus and to develop good policies. The thanks, first of all, to our partners here at the Baker Institute, Ambassador Judgeon, thank you very much. The founding director, Professor Neil Lane, Neil's down in front of me, and Dr. Kirsten Matthews, as well, who's a fellow in science and technology policy here. We've partnered, as May said, a number of times with the Baker Institute. We couldn't have a better partner for the sorts of things we do. Houston could not have a better public policy institute. It's a major contribution to intellectual life in Texas, I believe. Also, thanks to the Texas UK Collaborative, Dennis, thank you very much, and Dr. Gillis, who spoke in front of me, for their support and partnership over many years, not only for this conference, but for the many other things which Dr. Gillis alluded to. My thanks also to our two distinguished speakers, and I will get out of the way as quickly as possible, Professor Neil Lane and Lord Naren Patel, and I hope their presentations will create a good basis for discussion this evening. I believe we have a chance for some discussion after the presentations. And finally, and perhaps most importantly, our thanks to the sponsors, without whom this certainly would not have been possible, Stematics and the Huffington Foundation. We really are grateful for the support that you've given. And thank all of you for coming. And I should acknowledge in all of you, without wishing to show any particular favour, I'd like to say thank you to Representative Hardcastle, especially for coming and acknowledge his presence this evening. Thank you, Representative.
Thank you, Paul. Now we'll begin the program with our two keynote speakers. But just before that, I wanted to mention that during today, we really had an excellent conference in the morning organized by the Texas Collaborative on research on stem cell. And we hope that a lot of the collaborations that we saw come out of the last conferences will also continue to do so. There'll be more on that tomorrow morning if anyone's interested in attending at the same venue. And we also had a very interesting policy discussion this afternoon, and we were joined by very distinguished speakers for that. So I'd like to introduce our first speaker, Dr. Neil Lane. Professor Neil Lane is a senior fellow in science, technology, policy, and founder of the science technology program here. He's also the Malcolm Gillis University professor at Rice and a professor in the Department of Physics and Astronomy, his first discipline. Neil is well known for serving as the science advisor to President Clinton and also for being the director of the National Science Foundation. And his CV is really far too long to encapsulate in this small paragraph, but I know from people we work with all around the US that he sits on many advisory committees and boards and travels tirelessly within the US and worldwide to help people and advise on science and science policy. Neil also was provost and professor of physics at Rice before he went to Washington, and chancellor of the University of Colorado, and has had many distinguished medals. One of them is he's a fellow of the American Academy of Arts and Sciences, and he was awarded the National Academy of Science Public Welfare Medal recently, 2009, the American Institute of Physics Katie Compton Medal, and Association of Rice Alumni Medal. So, Professor Lane.
Thank you very much, May. I appreciate the very kind introduction. Certainly these past 8 years since returning to Rice have gone very quickly. It has been a wonderful experience having the opportunity to work with my colleagues at the Baker Institute all across the campus in the Texas Medical Center, other parts of Houston, including the British Consulate at General Houston, and across the state is really a special privilege. It's a great time to be working in policy. I'm not a policy expert. I've got some scars and bruises that I'll spare you tonight, but I want to share with you a perspective, and I very much appreciate the invitation to join Lord Patel this evening to talk about a policy issue that is of great importance to all of us who care about the health of our families, of our children, our neighbors, people across our countries and the world, indeed, medical research and the promise of stem cells. This evening I've been asked to review stem cell policy in the United States, especially changes that have occurred in the past 6 months with our new President, Barack Obama. I need to make an upfront disclosure, which you've already heard. I'm a physicist. I'm not a stem cell expert. The only thing I ever worked on that was wet was liquid helium. It was very cold, and besides that, I only did the theory on that, or some theory on that. So our stem cell expert here at the Baker Institute is Dr. Kirsten Matthews, whom you all have met. And so if I say something wrong about the science, she, as well as many of you here, can hop up and correct me. Since I am the Malcolm Gillis University professor at Rice University, with Malcolm sitting here, only a few yards away, I really don't want to muck it up, so a little bit nervous. I'll give you a little history and about the way policy has evolved in this country, and it may not appear to be a totally rational process. Ambassador Derigian is very familiar with the American process, policy making. I'm sure in the State Department it really was rational, but in the rest of government left something to be desired. Perhaps the Texas legislature would be a model of a rational decision making. But on a serious note, the UK, in fact, is a leader among nations in demonstrating that there really is a rational approach to setting policy. And I'm really, again, delighted that Lord Patel and, of course, Lady Patel could be with us to discuss this evening. So with the help of technology, I'll try to give you a picture of the situation in the U.S. If the slide will go. Yes. Now, in recent decades, a number of events, largely to do with advances in science and our understanding of human cellular biology, medicine, and technology, have brought us to where we are today. The slide shows the dates of some of the events that have influenced U.S. policy on stem cell research and, of course, many other related policies. Highlighted in blue are some key events that we just sort of arbitrarily chose because I want to talk about some of those. And there are other relevant occurrences that are represented up here in black. Now, the U.S. policy dealing with embryonic research began, as did many people's beginning to think about embryonic research, in 1978 with the birth of Louise Brown, the first test tube baby. And I will talk about some of these issues in detail. So For example, the Dickey Amendment, I'll explain to those of you who haven't followed that closely what that's all about. Dolly, we all read about. We know about the 1998, the creation of the first human stem cell line in 2001. We'll talk a little bit about the Bush policy. And then 2006, we'll talk about the Bush vetoes, which were perfectly consistent with President Bush's position on stem cells. And then try to bring us up to the Obama executive order and the NIH guidelines, and then talk a little bit about what might happen in the future. Now, when you map these events on, I want to do this. When you map this onto a timeline, you see that in this country, almost 20 years passed between the birth of the first test tube baby and any sort of serious consideration, serious discussion of policy having to do with embryonic research. You'll hear from Lord Patel that in the UK, there's really been more of a continuous discussion about these important issues and policies guiding such research. Now, the first event on my slide, the birth of the first test tube baby in 1978, really set the stage for the discussion in much of the world, including this country, of embryonic research. At the time, the regulation of such research in this country was governed by Title 45, Part 46 of the Code of Federal Regulations, in case you're taking notes. The Carter administration established an Ethics Advisory Board to oversee research on human embryos. Any research that the board approved could be carried out. The board met only once, as far as we know, never approved any research projects, and was disbanded sometime in the early 1980s by President Reagan, I believe, in part probably because of pressure from anti-abortion supporters, many of whom, at least at the time, opposed in vitro fertilization as well. Since it was no longer a board, then according to the law, no embryo research could be carried out. In 1993, during the Clinton administration, early in the administration, the law was rescinded, in theory allowing research on embryos to be carried out. However, as far as I know, no research was funded by the federal government. And President Clinton, being though very supportive of medical research, still wanted to address modern medical research developments, and therefore directed that NIH should be allowed to fund embryonic stem cell research, human embryonic stem cell research, with appropriate guidelines, oversight, and regulation. But the president was not willing to allow federal money to be used for the creation of embryos for research purposes. But I think the absence of a binding law on the books with appropriate guidelines and oversight and regulation to guide embryo research left a void that predictably was addressed a few years later by a Republican Congress using the appropriations process. One lesson here, I think, is that for us it's usually better to have a thoughtful policy on the books, a real federal law, than to take chances, as political winds can change very rapidly here in the U.S. And it's sometimes easy for the majority party, using this vehicle of a rider or an amendment to a funding bill, to make a major policy change that's very difficult for the president to veto. Sometimes the president will do that, but it's very hard. And that's just what happened. So, with the Republican takeover of Congress after the election of 1994, sometimes called the Gingrich Revolution, I remember it. I was at National Science Foundation in those days. It was a very interesting time in American government, is probably the statement I would make. Republican members of Congress were anxious to fill this vacuum, this policy gap on embryo research. And so the Dickey-Wicker Amendment, named after two Republican congressmen who introduced it, and more commonly known as the Dickey Amendment, was first attached as a rider or an amendment to the Department of Health and Human Services Appropriation Bill in 1996. 1996 was just an extraordinary year. It really was the first year the new Republican Congress could engage the White House on the federal budget. It was a very chaotic year, a very difficult year, and shut down the federal government for some period of time. Now, of course, the reason that rider controlled stem cell research was because the National Institutes of Health are part of, as most of you know, part of the Department of Health and Human Services. So that was the bill that controlled their funding. So the Dickey Amendment didn't say that you can't do this research in America. What it said was federal government cannot fund this kind of research. The amendment states that none of the funds made available in this act may be used for the creation of a human embryo for research purposes or research in which a human embryo is destroyed, discarded, or knowingly subjected to risk of injury or death greater than that allowed for research on fetuses in utero. The law specifically bans using the federal funds for the creation of human embryonic stem cell lines, as well as research using the nuclear transfer technique, somatic cell nuclear transfer. The wording also leaves open to interpretation funding research on pre-implantation, genetic diagnosis, or some of the alternative methods of deriving human embryonic stem cell lines, which could possibly harm an embryo. So there was some ambiguity then in the language. The Dickey Amendment then caused NIH and its parent department to go through a lengthy legal exercise to see if President Clinton's directive was allowed by the Dickey Amendment. They concluded it was, but by the time they've made that conclusion, jumping forward a bit, it was too late to set the guidelines, get the grants out the door, and then the change of administration came in 2001 with the election of President Bush. So meanwhile, science continued to move ahead of policy, but not without federal funding in this area. Another key event was the birth of this cuddly sheep, Dolly. Now, on July 5th, 1996, Ian Wilmot's group at the Roslin Institute in the UK created the first cloned mammal and named her Dolly, after Dolly Parton, for reasons many of you perhaps know. After the report of her birth was published in February 1997, Dolly's picture appeared in newspapers, magazines everywhere, TV screens all across the planet. And this event created a strong reaction among members of the public, as well as policymakers. Many people were afraid the next step would be cloning of human beings, very unpopular notion, and not only among members of the public, but also scientists. No reputable scientist, to my knowledge, has favored research to reproduce a human being through cloning. I might mention that by that time I was in the White House, and my strategy in the White House was to try to get people interested in science at an appropriate time in an appropriate way. And Dolly was a really good vehicle for that, because people in the White House staff were very interested in Dolly. So when Dolly was reunited with her mate, I've forgotten Dolly's mate's name, but anyway, then she became, in a family way, we sometimes say. And they loved all that, and they had children, and I just give regular reports on that along the way when business was light, when nobody had been assassinated or there were no wars breaking out or something like that. Also, the press was very interested, and so I gave a speech one time in the press club in Washington. Many of you have been there, given speeches there, and my wife was also invited, and she was sitting up front. And those guys are just really clever. So the last question, I took a bunch of questions, and the last question, which I thought would be a softball question, was, Dr. Lane, if you could clone anybody in the world, who would it be? And so I pointed to my wife and said, would you please stand up? Now, they thought it was a clever answer, but the more I reflected on the reality of having two identical wives, I'm not sure it was so smart after all. Following Dolly's birth and fame, President Clinton called on his Bioethics Advisory Committee, chaired by Harold Shapiro, at that time president of Princeton University, to give advice on what should be the government's policy. And the commission did make recommendations, in fact, rather quickly, on what they hoped the president would do. There was, as you know, there was no federal law at that time, nor is there today, that prevents someone from attempting to clone a human baby. Obviously, you can't use federal money for it in this country, but there's no law that prevents you from doing it. Fortunately, the Food and Drug Administration can backstop cloning research to a certain extent, and they promised to pay attention to this issue and regulate any efforts at human reproductive cloning. But it's kind of a dangerous policy situation for a country to be in. Well, the Bioethics Commission concluded that reproductive cloning of a human being should be prohibited at this time. And as a result, President Clinton put forward a bill for congressional approval that would ban human reproductive cloning. It's as far as it would go. Well, Congress, I think in retrospect, in a moment of wisdom, decided to postpone the passage of the bill so they could determine its impact on valid biomedical research. Also, the reason I think it was wise is several of the bills that were put forward during that period were so poorly worded that at least some of them would, I think, have prevented reproduction by any means whatsoever. So sometimes it's better Congress not act at all than to act rashly. Such a bill was never passed. In August of '98, after 5 years as director of the National Science Foundation, when I didn't have any dealings with biomedical research, I moved to the White House and had the experiences that I described a little bit earlier. In November 1998, James Thompson and his group at the University of Wisconsin-Madison reported that using non-federal funds, because that wouldn't have been possible because of the Dickey Amendment, they had created the first human embryonic stem cell lines from frozen in vitro fertilization embryos that were to be discarded. This breakthrough received a lot of attention by the media, and the general public began to understand that research on human embryonic stem cells could lead to new medical treatments, even cures, for some of the most debilitating diseases and injuries. I hasten to say that while this is a very beautiful stem cell, it's a mouse stem cell, not a human stem cell. So those of you who hear from the general public that mice are not beautiful and they're not wonderful, I'm going to show them this stem cell slide. President Clinton policy on stem cell research then was not really in place as far as he was concerned, and so he asked his Bioethics Advisory Committee to consider now Thompson's results and advise him again on what kind of policy we should have, not on cloning, which he'd already dealt with, but on human embryonic stem cell research. The Commission recommended that, in fact, the federal government should fund human embryonic stem cell research, including the creation of new stem cell lines from IVF embryos, and recommended steps to ensure appropriate oversight and regulation. Absolutely key. I know Lord Patel will emphasize how important the latter is. The Bioethics Advisory Commission, if you read that report, really didn't see an ethical distinction between the federal government funding research on stem cell lines that somebody else created by essentially destroying an embryo and actually using federal fundings to create stem cell lines. But the president did see a difference, and besides that, the Dickey Amendment was in place so that it would have been illegal to do so. With the president's okay, however, the Department of Health and Human Services and the NIH began to review that amendment to determine if the law would allow, as I said before, NIH to fund that research. They did, in fact, do that. They decided that the president's directive was consistent with the law, that NIH could do that funding, but unfortunately, the time did run out before anything could really be done. When President Bush came to office, he put a halt on any discussions of research on human embryonic stem cells until his administration had a chance to deliberate on the subject and determine give him advice on what he really ought to do. So I left the office with President Clinton sort of one day before the inauguration, and a lot of other Texans were coming to town. Neil was heading back to Houston. I actually wasn't invited to stay in the White House. I don't want you to think that I wasn't willing to help. On August 9th, 2001, President Bush announced a new federal policy set by his executive order. Federal funding would be available to support research only on stem cell lines existing before that day, August 9th, 2001. No funding would be available to create new lines or carry out research on lines that were created after the August 9th date, regardless of how they were created or by whom. The President announced, based on NIH information, that there were approximately 60 stem cell lines that would be available for research. It was overly optimistic. The number of lines turned out to be of the order of 21, not 60. Still, I think President Bush felt he was responsive to the scientists in allowing some funding for human embryonic stem cell research. Because to be fair, that's the first time the federal government had funded any human embryonic stem cell research, was in the Bush administration. But the response from the research community was mixed, many criticizing the new policies being too restrictive. They wanted President Bush to go further. I don't know why anybody thought he might go further, but there was disappointment. And I think that started a rift between the science community and the Bush administration that, along with a number of other happenings throughout the two terms, just made that deeper and wider in a very unfortunate way. Of course, as I said before, there are no federal laws that restrict human embryonic stem cell research or somatic cell nuclear transfer using non-federal funds, or even human reproductive cloning for that matter, should anybody be foolish enough to try that. Efforts have been made along the way to pass laws banning human reproductive cloning, starting with the President's bill, but many conservatives won't support such bills unless they go further and control other kinds of research involving human embryos. Of course, any human testing of new medical treatment or therapy is subject to FDA approval. So that's really the only backstop we have. Over the next 8 years, Congress tried each year to pass bills that alternated between expanding and prohibiting stem cell research. The first bill to come through both houses and go to the President's desk was the Bipartisan Stem Cell Research Enhancement Act of 2005, and then later 2007. The bill would allow federal funding of research utilizing human embryonic stem cells that were created from donated IVF embryos that themselves were created for couples who wished to have a child. After years of being pushed aside, the bill was finally passed by the House in 2005 and by the Senate shortly thereafter, within the following year. But it was vetoed by President Bush, who advocated embryo adoption programs. Well, it's estimated there are several hundred thousand embryos in IVF clinics all over the country, most of which will be destroyed because they are not needed by their donors or their prospective parents. Only a small fraction of this total, I think it's of the order less than 1,000, has actually been adopted. You may have a better number than that. In 2007, with the newly elected Democratic majority, the bill was again passed and again vetoed by President Bush. President Bush instead issued an executive order which clarified the meaning of risk to harm in his earlier executive order, so that alternate procedures to create human embryonic stem cells could be federally funded. Well, while the federal government has been unable to develop laws regulating human embryonic stem cell research, states have been much more proactive. Some states, highlighted in red on this slide, have passed legislation that restricts or bans such research. Other states have more moderate restrictions, highlighted in yellow. For example, Missouri has only banned the use of state funds for human embryonic stem cell research. It also passed a law allowing researchers to carry out this kind of research if it is federally fundable, basically protecting the research and the funding sources. In addition, several states, including California, Connecticut, and Illinois, have found funds to support human embryonic stem cell research in their state, highlighted in green. Majority of states, seen in gray, have no policy, and Texas is one of those. Now, when running for president in 2008, President Obama promised to increase federal funding for human embryonic stem cell research. Once in office, he moved quickly to change the policies of his predecessor. On March 9, 2009, President Obama signed an executive order entitled Removing Barriers for Responsible Scientific Research Involving Human Stem Cells. The order rescinded previous executive orders on human embryonic stem cell research and removed the date limit for federal funding of that research. It also directed NIH to develop guidelines within 120 days for the regulation of federally funded human embryonic stem cell research. Obama's executive order specifically states that the Secretary of Health and Human Services, through the Director of NIH, may support and conduct responsible, scientifically worthy human stem cell research, including human embryonic stem cell research, to the extent permitted by law. And that phrase, to the extent permitted by law, implicitly refers to the Dickey Amendment, which continues to appear on the annual appropriation bills for HHS, and its ban on creating human embryonic stem cell lines using federal funds. Obama did not intend for the new policy to remove the ban on using federal funds to create these lines, nor has he advocated for its removal. Many in the science community would like to see that eventuality. As requested, NIH produced draft guidelines for public comment in April 2009, and in the comment period, which is 30 days, over 49,000 comments were received on various aspects of the draft guidelines. Addressing the issues raised by the public comments, NIH then released its final guidelines on July 7th of this year. The guidelines were specifically written to address human embryonic stem cell research and certain uses of pluripotent stem cells. The guidelines highlight two basic principles for federally funded human embryonic stem cell research. First principle, responsible research on human embryonic stem cells will improve our understanding of human health. Second principle, individuals donating embryos should do so freely. What is eligible under the new guidelines? Well, for the new lines, only human embryonic stem cell lines created from leftover IVF eggs that were created for reproductive purposes and freely donated, and those lines for which proper consent and its documentation of consent can be provided. As part of the consent, NIH required that no payments or free services were provided in exchange for embryo donations, and that there is a clear separation between IVF procedures and the decision to donate. They also stressed that the donor was fully and completely informed, including notification that the embryos would be used to create human embryonic stem cells and that the embryos would be destroyed in the process, that the lines would be available for years, and that personal information might be provided to the researcher, for example, a family medical history or something of that nature. For already existing lines, NIH would review the documentation to determine if the investigators had followed the spirit and the intentions of the guidelines, and specifically that the investigators had demonstrated that the lines were created from leftover IVF embryos and donors gave informed consent. As well as highlighting what's eligible, the guidelines also outlined what is not eligible for federal funding. So what is not eligible? Human embryonic stem cells or induced pluripotent cells introduced into non-human primates, monkeys, for example. Not eligible would be breeding animals where the introduction of human embryonic stem cells or induced pluripotent cells may contribute to the germ line of a mouse or a hamster or any animal. Also not eligible, derivation of human embryonic stem cell lines, and not eligible human embryonic stem cell lines from other sources. Transfer, for example, parthenogenesis, the development of an embryo from an unfertilized egg, or any IVF embryos created specifically for research purposes. I think there's a number of places where the UK law differs from where we are at this time. When you compare the policies of the three recent U.S. administrations, you can see that President Obama's policy, at least on the surface, appears to be closer to that of President Bush than that of President Clinton. Although the Clinton administration never had a chance to fund such research because the clock ran out, the Clinton policy would have allowed NIH to fund research on human embryonic stem cells created from all sources, although it did not permit, as I said before, NIH to fund the actual creation of the lines. All three recent presidents agreed on that. So the line under can you use the money to create human embryonic stem cells is a no, no, no for the three recent presidents. The major difference between the human embryonic stem cell policies of Presidents Obama and Bush is the arbitrary deadline President Bush set, namely August 9, 2001, Obama's policy simply removes that deadline. In addition, Obama's policy more closely regulates the informed consent process and thus is somewhat more restrictive than that of President Bush. Turned out that several of the lines, human embryonic stem cell lines approved by the Bush administration, were found not to have proper documentation of consent, although federal funding had been allowed. So what do we have to look forward to in the United States? Well, alternatives to human embryonic stem cells are still being researched and could limit the need to create new lines of human embryonic stem cells, having an impact on current policy. Furthermore, politics is never over. Is it representative? Never over. In Washington or at the state level, and after President Obama's executive order expanding federal funding, many anti-stem cell groups moved their campaigns to the states, including Texas, to promote legislation to ban all human embryonic stem cell research or a portion of such research in some states, for example, to make sure no state funding goes for such research. And we could well hear more from Washington. The U.S. Congress still has not been successful in passing a law on getting a law on the books. Congress might propose its own policies and regulations governing federal funding of human embryonic stem cell research, or Congress might decide to restrict research being pursued by researchers using private or states funding. I don't expect that, but Congress surprises you sometimes. The lack of any federal law dealing with an attempt to reproduce a human being by cloning techniques, I think, is a very serious flaw in our policy. Now, given the breadth of other business in the coming years and the promise of alternative techniques, such as induced pluripotent cells that do not involve embryos, my guess is Congress won't spend a lot of time on embryo research policy unless something major happens that gets the public's attention. For example, a major cure that can be traced to human embryonic stem cell research or other research involving human embryos. I think that could make a difference. But there are many experts in the room, and you may have different views on the subject. So before I finish, I want to take a minute to tell you about our stem cell policy activities at the Baker Institute. The Baker Institute's International Stem Cell Policy Program is run by my colleague, Dr. Kirsten Matthews, molecular biologist. Our objective in this program is to encourage communication through events, research publications, web postings, reports, various stakeholders, with the hope that more informed policies will result. Our program advisors include Dr. Brooke Brody, medical ethicist, Rice Professor of Philosophy, and Director of the Rice-Baylor Center for Medical Ethics. This is a joint center. It doesn't mean Rice and Baylor are merging. I'm not announcing anything. The center already exists and has for a very long time. Thank you. Another of our advisors, Dr. Paul Simmons, University of Texas Health Sciences, Institute of Molecular Medicine, and UK researcher Dr. Stephen Minger, who often travels to Houston but was unable to attend this workshop due to a schedule conflict. And Bill Brinkley, who's Dean of Graduate School of Biomedical Research, Biomedical Sciences at Baylor College of Medicine, has been a key advisor from the very beginning of our program. We're indebted to him for his wisdom on this subject. We also want to recognize our many supporters for our stem cell program over the past 5 years, specifically the state of Qatar, who gave a generous endowment for our program, a science and innovation team, British Consulate General Houston, May Akrawi, Catherine Santa Maria, and of course the Consul General. The Richard Lounsbury Foundation, as well as supporters who've helped us over the years that include the Stematics, the University of Texas MD Anderson Cancer Center, University of Texas Health Science Center at Houston, Texas UK Collaborative, University of Texas Medical Branch, Texas Tech Health Science Center, Baylor College of Medicine. I do look forward to Lord Patel's remarks. Thank you very much.
Thank you, Professor Lane. And now it's my great pleasure to introduce Lord Naren Patel, who's the chairman of the UK Stem Cell Network and also the UK Stem Cell Oversight Committee. He's also chair of the steering committee of the Stem Cell Bank. And Lord Patel has been a long-term player in this area, and he was one of our first speakers here in 2004, I think, in our first program, and we were very, very pleased to have him again, because there's been a lot of change and legislation that he's going to tell you about since that time. And he's also chairman of the National Patient Safety Agency of England and Wales, Chancellor of the University of Dundee in Scotland, and his own practice was in high-risk obstetrics. He has several affiliations and is a member of the Royal Society of Edinburgh, honorary fellow of several royal colleges in the UK, Ireland, and overseas, fellow of the Academy of Medical Sciences, holder of several honorary doctorates in the UK and abroad, and he received his knighthood in 1997 and was elevated to peerage in 1999. And Lord Patel sits on the Science and Technology Committee in the House of Lords, where our team interacts with him frequently. So we really thank him for coming all this way to speak, and I'll introduce him.
Good evening, ladies and gentlemen. It's for me nearly 2 o'clock in the morning, and I thought I might wake myself up. So when coffee came around, the lady asked me if I wanted some coffee, and I said, as long as it's not decaf. And to my surprise, she said, all coffee here is decaf. If that had been so in Europe, there would have been a riot by now. So I may fall asleep. And I was glad to hear that we had some new recruits this evening, because those of you who have stayed the whole day here already have heard much of what if not all, of what I have to say. So please excuse me if this is a repetition to you, but maybe you'll spot some differences in the presentation. So I'm going to talk mostly about United Kingdom legislation related to embryonic stem cell and embryonic stem cell research. But I emphasize that whilst all of the talk is related to legislation and policy related to embryonic stem cell, we promote actively research in all types of stem cells, adult, cord blood, fetal, and embryonic, because at this stage we cannot be sure which of the ones will deliver the treatments, and it is likely that all of them will deliver treatments, but of a different nature and of different diseases. Secondly, that all of the legislation and regulation is underpinned by ethical debates and ethics in the first instance. So ethics has driven, I hope, our formulation of legislation and the regulation that we have put in place. We have in the United Kingdom regulation related in vitro reproduction of human embryo research, of human embryonic stem cell research, human tissues, and also have governance of stem cell lines, particularly human embryonic stem cell lines. And today, unless we have time, I will not mention what governance we have in place related to embryonic stem cell lines, but this is related to every line created in the United Kingdom to be deposited in the bank, and an independent steering committee adjudicates as to how these lines are used. So if I go back to these two gentlemen, and the one on this side is Patrick Steptoe, and that's Edwards. This is the day they announced to the world the birth of Louise Brown in London. It was actually announced in a hall in the Royal College of Obstetricians and Gynecologists, and it so happens that the hall is called Nuffield Hall because the donation by Lord Nuffield, who did not have any children, also happened to be distant uncle of Patrick Steptoe, which is quite remarkable. Following that, there was much anxiety, particularly in the press, that as in vitro fertilization baby was born following in vitro fertilization, labeled the test tube baby. There were all kinds of anxieties that will now have monsters created in a dish that will then become fully grown human beings. That led to to establishing a voluntary licensing authority, which was established by the Royal College of Obstetricians and the Medical Research Council. Now, one thing we have in the United Kingdom is if my professional organization, the Royal College, gives me guidance of how I must practice, in most of the cases I'll obey it, because penalties sometimes for not obeying can be quite serious in terms of your professionalism. So the voluntary license authority worked well, but there was a need to have further look and need for legislation. And Warnock inquiry was set up into human in vitro fertilization, social, ethical, and legal implications following the birth of Louise Brown in 1978. The inquiry reported in 1984, and in 1990 it led to the establishment of Human Fertilization and Embryology Authority, commonly known in our country as the HFEA. That was established in 1990. By that time, Lady Warnock became Baroness Warnock, and she now sits in the House of Lords alongside me, and as she gets older, she gets even more and more liberal in her views. Quite the opposite to most of us who get older, we get more right-wing in our views. However, she contributed to that initial debate a lot. In fact, she probably was the one person that was instrumental in driving the legislation that we now have. The legislation at that time was focusing more on human fertilization and embryology, that we have in place regulation of in vitro fertilization. So, for instance, today we can tell exactly how many human embryos across the United Kingdom are in storage, how many were used for fertilization purposes last year, how many pregnancies resulted from every institution that provides that service. The use of gametes and storage of gametes is also controlled by the HFEA. It therefore carries out licensing of centers for reproduction, control of human gametes, monitoring of services, advice related to practice, develops codes, has a data collection system and monitoring so they can monitor poor practices or poor outcome institutions. And it does not involve in granting research licenses, but as far as embryo research is concerned, as I will mention in a minute, it has a greater role. So the Act of 1990 was about creation, keeping, use of embryos outside the human body, the storage and use of gametes. But there was an absolute prohibition of placing of non-human embryos or gametes in a woman. That was the first piece of legislation in 1990. And the research license can only be issued by the HFEA if HFEA was satisfied that the research is necessary or desirable for the purposes of promoting advances in fertility treatment, increasing knowledge about the causes of congenital disease, increasing knowledge about causes of miscarriage, developing more effective techniques of contraception and conception, developing methods for detecting the presence of gene or chromosome abnormalities in embryo before implantation. However, there was a rider put on that additional purposes may be added by affirmative regulation, but must be related to increasing or applying knowledge about the creation and development of embryos or about disease. So it was much more about improving fertility treatment than it was about embryo research. But what it did say was that embryo research was permitted. Further legislation was amended in 2001, which was about further purposes for which a research license may be authorized by the HFEA. The authority may issue a license for research under these schedules, and license may be issued for the purpose of increasing knowledge and about the development of embryos, increasing knowledge about serious disease, or enabling any such knowledge to be applied in developing treatments for serious disease. But for the first time, we also introduced complete prohibition of reproductive human cloning, with very severe penalty or long periods in imprisonment. So human cloning, reproductive cloning, was criminalized. And I believe we are one of the very few, if not the only country, that has this in legislation forbids human cloning, reproductive cloning. There are other restrictions in all human embryo research in the United Kingdom, and that is a 14-day limit on embryo research or appearance of the primitive streak. So no research can be carried out on an human embryo beyond 14 days. Embryos should not be specifically created for research unless the HFEA is satisfied that, and the word spare is a wrong word, embryos that are created for purposes of infertility treatment are not used and and are not available. So yes, the authority can license the creation of human embryos for research, but only if it's satisfied that there is no other means of doing that. To give you some idea of how many embryos might be available, this is over a 5-year period that about 760,000 embryos were created, 350,000 used for infertility treatment, 183,000 were stored for future treatment, 48,000 were given for use in research, so they were donated for research purposes, 118 created in the course of research, and 237 were not used for any purpose and were destroyed because they were no longer required by the couple for infertility treatment. So at the time that this legislation was going through to allow the procedure for creating embryos using the somatic cell nuclear transfer, the House of Lords only allowed that legislation to go through on the condition that a select committee will be set up of the House of Lords that will look at the ethics of embryo research and stem cell creation, a status of early embryo, that will take the views of the faiths, all faiths in the United Kingdom, and the social and legal context. That select committee was chaired by Bishop of Oxford, and those of us who spoke for embryo transfer were not allowed to be members, neither were those who were directly opposite views about it, opposed it. So the principles they came out underpinning the use of embryos in research are that the embryo of the human species has a special status, but not the same status as a living child or adult, that the human embryo is entitled to a measure of respect beyond that accorded to an embryo of another species, that such respect is not absolute and may be weighed against the benefits arising from proposed research, and that the embryo of the human species should be afforded some protection in law. And that underpins all our regulation related to in vitro fertilization and human embryo research. And whilst it could not define, as James Lawford Davies said earlier on, as to what that respect should be, it defined it in terms of regulation that protects the use and defines the boundaries that cannot be crossed when you use human embryos for research. So embryonic stem cells in 2001, the legislation allowed such lines to be created, including somatic cell nuclear transfer, the technique that was used to create Dolly the sheep. But there was still imposed a 14-day limit on embryo research. Embryos should not be specifically created research, only if spare embryos cannot be used. And that legislation, again, was put in place. So this is just showing you that early embryo formed in in vitro fertilization, the early embryos formed by cell nuclear replacement, umbilical cord or fetal tissue, and adult cells. All of this research and all of these different types of stem cells is now permitted in the United Kingdom. That's a picture of human embryonic stem cells about the size of a small grain of sand or a pinhead that can now differentiate into all the 257 different cell types that exist in our body. So embryonic stem cells from in vitro fertilization, the key steps are the fertilization and harvest of embryonic stem cells from an early blastocyst. We allow, through legislation, if a license is given by the HFEA, embryonic stem cells from cell nuclear replacement techniques. And the reason we allowed this was to make it possible to create stem cell lines that carry the disease and the genes that the affected person has of that disease, and to see if it's possible to have stem cells extracted from the blastocysts and the stem cells made to specialize, for instance, into nervous cells or heart muscle cells or insulin-producing cells, and these cells are a perfect match for the patient, so it does not have the immune problem. That's the research basis why we allow creation of embryonic stem cells using the SCNT technique. However, our chief medical officer in the year 2000, in a report, Stem Cells Research, Medical Progress with Responsibility, in his annual document said that the technique of using animal egg as shell to carry human nucleus, most researchers active in this field do not consider this a realistic or desirable way forward. However, that had changed by the year 2006, and the scientists were already talking about possibly using some form of animal-human hybrid embryos to be able to create embryonic stem cell lines that carried the disease genes to be able to do research. The main reason scientifically for wanting to do this is the lack of human eggs to create such lines. That the human eggs has a problem, and it causes it causes a lot of not only hardship, but could cause complications to a woman. And that if it were possible to use an empty shell of an egg from an animal and use nucleus from a somatic cell nucleus from a human to create an embryo, this will be fundamentally 99% at early stage be human DNA, and that such embryonic stem cell lines will behave like a human embryonic stem cell line to allow research. So this is a picture of a hybrid or a chimera, and this is a real picture of a hybrid cell. So last year we discussed the legislation, whether we should allow this or not, and the Parliament came down on the side of allowing further human admixed embryos to be created, an interspecies embryo and to be created. About that same time when we were debating this, the news broke about the induced pluripotency of the adult cells from Japan using some viral vectors. And as we all know now, IPS, or the induced pluripotent cells, is a possibility. Nonetheless, we felt that at this stage was still advisable to allow the using of human admixed embryos. The legislation that defines says they're allowed as long as it's the human DNA that predominates, because it was difficult to decide in legislation what percentage of DNA should be human. Most of these embryos, and the scientists were only wanting to use cytoplasmic embryos, where the human DNA will predominate by about 99% at the stage when stem cells are harvested. So we do allow interspecies embryos to be created and used for research purposes, all of these different types. So the review of the 1990 Act and the public consultation in 2005, a white paper in 2006, and the HFEA Act November 2008. And the important part here is a public consultation. At every stage of our legislation, and you already probably spotted that our legislation is progressive, and it moves with the need to have, as science advances, the need to have the legislation to protect public confidence. And at all stages, our research councils conduct a wide public consultation. And hitherto, every time a poll is taken, and the latest one was taken November last year, about 70% or more of the public is fairly relaxed about the legislation for two reasons. One, they understand what's trying to happen, what people are trying to do. Two, that we have a regulatory authority who is responsible for making sure that there is no abuse by scientists. And scientists also know that the penalties for getting outside the boundary of the legislation is pretty severe. The other three parts of the legislation, or two other parts were the parenthood of a child and miscellaneous and general provision, but the main part was the amendment to the Act 2009. when it took account of the scientific developments, reflects change in social attitude, regulate according to principles of better regulation. So it allows now, includes embryos without fertilization, human admixed embryos, gametes to include primary oocytes and spermatocytes, defines embryo, eggs, sperm, and gametes, so that only those permitted gametes and permitted embryos can have research carried out, and the nuclear DNA to include pronucleus. I'm sorry, something's happened here. Okay. There is prohibition of placing of embryo in a woman. The only embryos that are allowed to be permitted to be placed in a woman are so-called permitted embryos using permitted gametes. Prevention of transmission of serious mitochondrial disease is also permitted, and genetic modification of cells, embryos under research license by HFEA only is allowed. Now, why did we allow the mitochondrial disease embryos to be created for study of mitochondrial disease? Mitochondrial disease is chronic, progressive genetic disorders. The incidence is about 1 in 3,000 to 4,000, and nearly 40 different types of serious diseases occur as mitochondrial diseases and are inherited through the mother. There are more than 100 such diseases, but nearly 40 are very serious. So what we allow is to use couples fertilized egg and pronuclei removed and introduced into donor egg cytoplasm, from which the pronuclei are removed. So you end up with an embryo with donated cytoplasm containing normal mitochondrium. This is to try and eliminate these mitochondrial diseases. Some people refer to this as an embryo with 3 parents, because 3% or less of the DNA has come from the cytoplasm of the donor ovum. There is, as I said, stem cell governance. So all lines created using human embryos, human embryonic stem cell lines, the law demands that all such lines be put into the stem cell bank. The stem cell bank curates these lines and also does research on it to make this characterize these lines, so these lines are better for research purposes. Scientists can obtain these lines, but the steering committee has to be satisfied that the research they want to obtain the lines for is bona fide research and is approved. The committee is independent. Although it is chaired by me, it has theologians, ethicists, moral philosophers, and 1 or 2 scientists on it. They are in the minority. But majority is lay people with background in ethics, law, and in theology or science or ethics. The most important part is the dialogue with the public, that we engage the public about development in stem cell research, identify a range of views about science, ethics, societal context of stem cell research, raise public awareness and opportunities and challenges and uncertainties of stem cell research, and long-term public dialogue. And last year, the biggest exercise was carried out using over a period of 18 months called Science Dialogue with the Public. And the public, again, 70% of the public felt very comfortable about the legislation. The other important contributors are the disease-based charities, the Parkinson's, the Alzheimer's, the cancer, different cancers, the diabetes, and other disease-based charities who have always backed this legislation because it is well regulated. So in summary, our legislation is sometimes said it is liberal, but it isn't. It is highly regulated. Not only is the research regulated on human embryos, the whole area of in vitro fertilization is regulated. And research can only be carried out if the authority gives a license, and it has to be satisfied that the research cannot be carried out by any other means, that the research can be carried out without using human embryos, that the research can be carried out using the embryos that might be available that are not required for infertility treatment. If all of this cannot be done, only then it might grant a license to do research on human embryos or create embryos for research. Since 1991, 124 projects have been granted by the authority. In the year 2008 and 2009, 34 licensed research projects were granted, 15 for the study of derivation of lines, 1 using the somatic cell nuclear transfer. 3 embryos were allowed to be created using human and animal eggs, 11 to study embryo development, 7 to study pre-implantation diagnosis, including 2 for mitochondrial disease, 1 to study maturation of eggs in laboratory. So over nearly 10 years now, there are not large number of licenses granted. Thank you for listening.
Thank you so much, Lord Patel, for giving such an excellent overview of the UK legislation and history. And thanks to Dr. Lane for his overview of presidential policies and where we are now. I think the ideal situation is that we end up with an international stem cell framework, an international stem cell bank that is regulated enough so that the public have confidence in it, but that also allows the research to progress to the therapies that it holds potential for, and really allows the best researchers in the UK and elsewhere to collaborate with the best of the best here in the US, and a lot of them are here in Texas. Before I turn it back to Professor Lane and Lord Patel, I just wanted to thank a few people whose names have not been mentioned but have really been instrumental in putting this 2-day event together. And I know we've mentioned Dr. Kirsten Matthews and Dr. Dennis Heaton, and I want to thank them again. Catherine Santamaria, who's worked very hard on this all summer, and also Melissa, Alex, and Whitney from the Baker Institute. Thank you all for everything. And not to forget Lisa Ross, who's worked very tirelessly to her, I don't know, is it over 40 scientists together over these 2 days? So something that very few of us would be able to achieve with this level of organization. So I'd like to invite Dr. Lane. Dr. Lane and Lord Patel, and open it to Q&A, which Dr. Lane will moderate.
I think there are microphones wandering around the room. I don't actually see any, but yes, there's a microphone over there. So please, if you'd like to, okay, and one over here, thank you. So if you'd like to ask a question, hold up your hand and somebody will swiftly bring you a microphone. Please. I don't think I need a microphone, but... Well, some of the people might have trouble, and besides that, I'm very old.
When do you really think we might see the commercialization of some of this research in a massive scale commercialization?
Do you want to comment on commercialization?
Well, not a field that I would regard myself as expert, so I will parrot what was said in an earlier discussion. I don't know if I see Chris Mason sitting somewhere. There he is, yes. And if I get it wrong, my parroting of what he said, then he'll correct me. Apart from adult stem cells, which there have been several successes, and the one that he particularly mentioned that I think was impressive. Chris, why don't you just tell them instead of me?
Thanks, Neil. Yes. If
Stand up in my behalf.
Is that okay?
Please.
If we look at the current translation and commercialization of regenerative medicine therapies, I think we have to, first of all, take into account there is something like 250,000 patients have already received living tissue-engineered skin successfully for chronic leg ulcers. Something like another 500 patients have received adult stem cells for corneal damage to the eye that would otherwise be permanent, with something like an 80% success record. And then we have the sort of therapies which are coming online now, such as tissue-engineered windpipe, trachea, for example, and the first patient was done in Barcelona approximately a year ago and is doing extremely well. So we're having successes with somatic cells, good numbers, probably 250,000 to 300,000. At least 500 patients have received adult stem cell therapies for the eye. If we don't count things like bone marrow therapy, which obviously are very successful for cancer therapies. And as we move forward into embryonic stem cells, we see Geron, who are still presently on clinical hold for their spinal cord injury, but moving forward. We see Novacell looking at diabetes. We see Paris, a group looking at embryonic stem cells for heart failure. In London, of course, Pete Coffey looking at age-related macular degeneration using embryonic stem cells for prevention of age-related macular degeneration. So things are moving forward very quickly. I think the important thing to note is that adult stem cells have been around about 40 years, embryonic stem cells 10 years. So where they are in their respective lifetimes, they're both well on track. Thanks, Darren.
Thank you, Chris. I thought you would say it better than you did.
That's just what you were going to say.
Yes, exactly.
Are there other questions?
I think the two important points, but because we're talking mostly about embryonic, is what Chris mentioned, that the Geron Corporation's spinal injuries trial will maybe soon go to clinical trial, but Coffey's in London is about to go to the trial for age-related macular degeneration. And those of us in the room who are over 65 years of age, it is known that in that age group, the incidence is 30% for age-related macular degeneration. So if it works, we're talking about tens of millions of people that could be treated.
And I think it's important. It's important to keep in mind that in just speaking about this country, there are many clinical trials of various kinds that don't work out, and undoubtedly that will be the case with stem cells as well. And however, there's because there is such great sensitivity on the stem cell issue. If something doesn't work out or something goes badly wrong, that could have very far-reaching consequences. And so my urging of our policy makers is sort of keep that in mind and be prepared when to respond and to deal with perhaps even public outcry under such circumstance. Also, this business of stem cell tourism that we talk about going on in various parts of the world, totally unregulated by who knows who. Some such incident could go terribly wrong, whereas all of us, I think, and I think we sort of have to be ready if that unfortunate event should happen. Please.
Yeah, Professor Lane, this is a question for him first. And you mentioned something in your presentation that you had said that what we really need to try to drive the point home for stem cell research here in the United States is a cure. And it's a four-letter word that a lot of us are really fearful of in biomedical research and in clinical practice and those kinds of things. And so short of that, and hearing some of the timelines that have been spoken already as to what we can expect, what are the kind of interim goals do you think that we could have that we could use to counteract a lot of the negative commentary that's in the blogs and the press and in these special interest groups that have really denigrated stem cell research down to the point right now where the common verbiage in there is there has been nothing from it so far, so why should we have it?
I think it's a very good question, and I don't have a strategy that I've thought through. Certainly, I think communication of accurate information in a dispassionate way with the general public is always a good idea, and that's fundamentally what this place is all about in many areas of public policy, including this one. I think, of course, it's important not to for scientists not to overpromise what any particular discovery or technique is going to deliver. I think it's important while adult stem cell research and the therapies are being advanced for the sort of community to have an appropriate story that is larger than this single narrow embryonic stem cell issue, but appropriately includes the importance of that. I think sometimes the public is quick to think a stem cell is a stem cell is a stem cell, and on one side that might mean if there's a great breakthrough with adult stem cells, that could translate in terms of public opinion more broadly, and I think generally it would. And then the opposition of human embryonic stem cell research would have to tool up to counter that, which they undoubtedly would do. But I think telling a straight story, especially guided by principles such as those that Lord Patel put up on that slide, and having a rational discussion where we're not yelling at one another, where when we get together in a room, we try to see what it is we all believe in, what we all agree upon, the importance of human life, the sanctity of human life, and then if there are issues about which we don't agree, see if one can have some discussion about that. My good colleague, Dr. Brinkley, here is a great expert at that, because I think, Bill, am I right? You're an East Texas man, and you still, I think, talk with churches and communities to a number of people who have different views from those of your own on this subject, I would guess. And I don't know, you might, if you'd be willing to recount an experience or add to what I commented, I think it might be very helpful. What happened to the microphone? Could we have a microphone here for Dr. Brinkley, please?
Thank you for mentioning that. I think that the biggest surprise I've had in doing this, going around the city, speaking to different church groups and civic groups, has been the welcome that I've received in every case. Very little criticism, very little argument. Thank you for clearing things up, and it's been a pleasure. And so I've really enjoyed it, and I think the best thing we can do is to speak to community groups, tell our story, try not to overstate it with premature hype, and you'll find out that under those conditions, you have the best of responses. The wars and fights that go on in the legislation amaze me, and I'm amazed that UK can get this much done without a lot of angry citizens and a lot of angry politicians always in the newspapers complaining about the evil scientist. I don't know what the difference is in the culture, but I'm wondering about, we did win our independence from those folks in England. I think they might have won the stem cell war.
If I read my history right, it did make a king very mad.
Other questions, please. Got the mic.
First of all, I want to thank you very much for a very good day. I did miss the morning not being a scientist, but I was here for the afternoon, and it was very educational. And I've been studying this since 2003, full-time as an advocate. But I do have one question about a subject that has, to the best of my knowledge, hasn't been mentioned yet, and that is with regard to pluripotent stem cells, disease in a dish, being able to create a disease, watch it develop, treat it with pharmaceuticals. From what I read, they're doing it. I'm not a scientist. I'm not reading all the same journals that all of you are reading. Does this hold promise for the future?
I hope I can illustrate by telling you the exact story the exact analogy that I used at the time when we were debating this legislation last November. And the analogy I used was because just then the news had broke that Yamanaka in Japan and others had produced induced pluripotent stem cells that behave like embryonic stem cells. And the analogy I used was, we have found a yellow metal that looks like gold. And the initial studies on it suggest that it could be gold. But we're still not sure. And the only way we'll know is to compare it with gold. So we do need to understand the biological behavior of the induced pluripotent cell that is exactly the same as the biological behavior of truly embryonic, human embryonic stem cells. If that turns out to be so, and in behavior, in all other respects, turns out to be so, then we won't need to use any more human embryos.
My question really wasn't that, because I said pluripotent, I meant embryonic and I meant embryonic in IPS. I'm asking about creating the disease because of pluripotency, whether it's embryonic or IPS, creating a disease in a petri dish and being able to study that disease, being able to treat that disease, and conceivably cut the cost and time on reaching conclusions for, if not cures, at least treatments.
If I understand you correctly, and I'm not so sure that I'd have, but if I do, I think you're saying that the embryonic stem cells and the induced pluripotent cells in a dish allows us to create stem cell lines that carry the disease genes that will allow us then to study the behavior and the progression of that disease and even find cures using drug or to alter or mitigate that disease, then the answer has to be yes, that is one of the possibilities too.
I think that we hear far too little about that, or the public hears far too little about that.
Yes, and to that also add being able to study drug toxicity
right.
in a disease line, stem cell lines, as opposed to giving it to human volunteers, as it happened 18 months ago in London, to youngsters who then developed serious conditions because of reactions to the drug that we were testing on them.
Thank you. It's a very important point, and it is one of the arguments why somatic cell nuclear transfer, I think, might be very valuable as a diagnostic tool, as a research tool, in the way you described, because you can control the DNA, you can take the DNA from somebody who's a victim of a terrible disease and do just what you described, if I'm correct. Other questions? Ambassador Georgian.
Thank you. I'm rather impressed by the success the UK has had in bringing public opinion forward on this issue. What do you attribute it to? Was it merely the legislative process, or was there a very coordinated public campaign, a public relations campaign to bring along public opinion?
I think it's a continuous public dialogue with the public, and explaining with the scientists, with the public, and sometimes legislators too, but mostly scientists and the public and others in the same room, discussing with each other and explaining to the public where science wants to go and why. And then having a discussion about what kind of regulation we can put in place that will make sure that it can be controlled. And if you ask the scientists, it is not easy to get a license. The authority is pretty draconian in the way it behaves and demands from the scientists all kinds of explanations. It also monitors, once it gives the license, it monitors regularly about how that research is going and what results they're obtaining, and are they still sticking to the research they said they will do. And it's answerable, the authority is answerable to the Parliament and to the public, so he's very aware of that.
Maybe take one more question, Representative Hardcastle.
I want to go back to the answer Dr. Bill gave a while ago, because all of y'all realize I'm not used to talking in front of this many scientists. I would a whole lot rather talk in front of scientists some days. But I think our greatest misnomer or our greatest misuse of what we got is falling into the opposition's trap of not being factual. And, you know, that's why I've been here for the second time, and I still can't quote science. But each time it gives me a little better understanding of how to argue or how to have that talk like you were talking about, have it on a Christian, faithful level and still make sense and maybe not be as passionate, but be factual and not start in with a lot of our opposition. You can't control what they say back to you, but it's generally 100% wrong, you know, and it's 100% emotion or it's 100% religious based or whatever, but it's even wrong at that. So I think as each of us go out of here, look for those places, those Lions clubs or Rotary clubs or youth groups or whatever, to explain, you know, that science is new. And, you know, I'm honored to be sitting here with all you smart guys and gals, but it's a new science. There's nobody in here that's an expert completely on the stem cell and where we're going on stem cell. But it's the promise that's out there.
I appreciate your comments very much. We hope you'll keep coming as well. One thing I feel very strongly about is that, and I think Bill Brinkley has demonstrated that with all the good work he's done in the community, is if we expect the public to listen to us, we need to respect each individual person, whatever their views are, wherever they've come from, their background. They're very important people. Whatever your own religious beliefs are, or I think from a perfectly pragmatic political point of view, you're not going to make any progress with any culture, with any group of people, unless you do that. If they feel, when you come into the room, that you're interested in them and their welfare, you care about their opinion, even if they know you're going to disagree. My own sense is, my own experience is, you've got to start there. If you don't have that, then you're not going to make any progress as well. Malcolm.
Yeah, Neil, I just wanted to add, I think in answer to your question, Ed, the scientific community in Britain pays more attention to this than we do in this country. They have been very upfront and proactive on the question of genetically modified food. The rest of Europe is absolutely crazy on this question, not in Britain. A better example is the report by the Royal Academy on Nanotechnology about 3.5 years ago. They directly faced all of the claims, some of the mythical claims, and some that had some basis in fact, about dangers in investing in nanotechnology and nanoparticles. And they covered it very well. They expressed their views very well. And as far as I know, this has pretty much quelled nervousness over nanotechnology in Britain. So they work hard at it, and that's what we have to do, and that's what these kind of conferences are all about.
Thank you, Malcolm. I think we're out of time. Are you going to close out? Or? Well, then I just want to thank Lord Battelle for this session and everybody else for this wonderful evening, sir. If there's no further business, there's still eats on the table. You have to finish all of these chocolate things and other things. Thank you all very much. Have a nice evening.